Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Today is part 4 of my series on sexual reproduction and inheritance. And of course, if you're going to start inheritance, you have to start with Gregor Mendel, because he's considered to be the father of modern genetics. And yes, I'm still sporting the hat from Frozen. I hope that years from now, my daughter will be proud of me for wearing a Frozen hat on YouTube. But hey, what can you say? I like that song, Let It Go. It's a good one. So why is Gregor Mendel so important? And the reason why is because he provided the first rules of inheritance. Up until his time, nobody really understood inheritance. And it was a big deal because he disproved the modern theory of the time, which is known as blending inheritance. And we're going to come back to that because this experiment provided major support for Darwin's theory of evolution by natural selection. And ironically, Darwin published in 1859, and for the next several decades, his theory was defended. And by the 1890s, his theory was actually on the downside, like people were believing in it less and less because of blending inheritance. And ironically, Gregor Mendel was doing his experiments in the 1860s when Darwin could have really used the results of his experiment, except Darwin was over in England speaking English, and Gregor Mendel was an Austrian monk publishing in German. So Darwin never read his manuscript, and in fact it was lost until about the 1890s when it was rediscovered, and people immediately applied it to Darwin's theory of evolution by natural selection because he disproved blending inheritance. I will come back to blending inheritance. Now, he did provide these first rules of inheritance by doing a brilliant experiment, and I'm going to talk about that. Even though it's been 150 years, it was groundbreaking and it's still relevant to this day. But I will say this, Gregor Mendel, he got lucky. He got really lucky. Really lucky. But he's also a good experimenter as well. But he got lucky. I'll explain. So most of us have had some type of high school biology, and we've come across Gregor Mendel in his really famous pea plant experiment, where he had these purple flowers and these white flowers, and they were considered true breeding, which means if I breed a purple flower with another purple flower, I always get purple flowers over and over and over again. But in pea plants, flower color is not the only trait he experimented on. He also looked at seed color and seed shape as well, and seeds could be yellow or green. So in his experiment, it was quite brilliant, he crossed true breeding plants that were purple or white flowered or green or yellow seed. And what he found was pretty cool. In the first generation, he got all yellow seeds or all purple flowers. But he didn't stop there. He did something really cool next. He bred a second generation by breeding that first generation together. And what he found was approximately 75% of the offspring had purple flowers, 25% had white, or 75% had yellow seeds, and 25% had green seeds. Wait, the green, that skipped a generation. What happened here? And this was a 3-to-1 ratio. 3-to-1 ratio, things skipping generations, that's important. Okay, so here's Mendels Interpretation. Now, this is where I say he was intuitive, but he also got a little lucky as well. This is one of the first places he got lucky. He knew nothing of the chromosomal basis of inheritance. You see, our chromosomes carry our genes in our DNA, right? Our chromosomes are our DNA, carries our genes. He knew nothing of this. That is kind of interesting to know that he came up with these laws of inheritance without knowing how these things were being inherited on the chromosomes. So what he realized, this is a breakthrough, was that each parent is contributing a heritable feature, a gene. So each parent is contributing a version of that gene known as an allele. Okay, so one parent is contributing a yellow allele, one parent is contributing a green allele. Think about flower color. One's contributing a purple flower color, the other one's contributing a white flower color. Now, what he realized, that was a pretty big jump. And then he realized that you can have two of these alleles and be the same. you're homozygous because homo means same. So if you're carrying 2 purple flower alleles or 2 white flower alleles or 2 green alleles, you are homozygous. Likewise, if I've got a purple and a white allele for flower color, you're heterozygous, or a green and a yellow allele, you'd be heterozygous. And if you're out there in your AB blood, guess what? You're also heterozygous as well for blood type. So heterozygous means I've got my gene, but I've got both versions of the alleles, or 2 different versions of the alleles. And then he realized, hey, wait, I crossed a yellow seeded plant and a green seeded plant, so my F1 were all heterozygous. They had 1 allele for yellow, 1 allele for green seed color. So to improve our understanding here, let's talk about genotype and phenotype really quick. Genotype are your actual genes. So the F1 generation, they were heterozygous. They were carrying the yellow and the green allele. And then you have your phenotype. That is your expressed genes. They were all yellow, so they were only expressing the yellow allele. Likewise, if you have purple and white flowers, the genotype for the F1 would be 1 allele for the purple flower, 1 allele for the white flower. That would be their genotype. And their phenotype would be purple flower. So then he's like, these alleles, they can be dominant. So the yellow allele is dominant. All you need is 1 copy of the yellow allele and you have yellow seeds. Same with the purple flower. If you get an allele for the purple flower, you have purple flowers, even if you have the white allele. In humans, if you get an allele for A or B blood, you will express that allele. Those are dominant. And then, of course, recessive. White for the flowers would be recessive, which means you have to be homozygous for 2 white alleles to express it. And green for the seed color was also recessive. Now, he also realized that flowers produced gametes. It's pollen for the males. If you've ever looked at a flower, you've got an ovary. Plants have male and female flower parts. So they realized that the males and the females are both producing gametes, and each gamete carries a copy of the gene of that particular allele, and you get one of those from each parent. And when those gametes are segregating, they do so independently during gamete formation. So if you're heterozygous for purple and white, you're going to separate them out equally. They're not going to have any effect on each other. And another law he came up with, and this is a tricky one for me because I don't like calling it a law, but it actually is, because if the genes are on different chromosomes, then they will separate independently of each other. For example, if you have a gene for flower color and seed color, and they're on different chromosomes, they will be inherited independently of each other. And when Mendel was studying pea plants, he got lucky. Most of the genes he was studying, like flower color, seed shape, pod color, most of those were on different chromosomes. So it looked like seed color and seed shape were inherited independently of each other. They had no effect on each other. Turns out that wasn't exactly the case. It turns out that some of these genes were actually on the same chromosome, but they were opposite ends of the chromosome. So if you watched my video on how meiosis generates diversity, crossing over would break up those combinations of those genes, but almost to look like they were independent assortment, but not really. So his numbers actually didn't come out to a perfect 3-to-1 ratio or a 9:3:1 ratio for 2 genes with 2 alleles each. They were close, and he did thousands of these breeding experiments. They didn't exactly come out because they were linked, but he's like, that's close enough. If the genes for like seed color and seed shape were close to each other on the same chromosome, he never would have come up for the law of independent assortment because they would have been inherited together. Who I think of as the actual father of modern genetics, Thomas Hunt Morgan, was really important because he actually figured that out, and that will be in my next lecture. So Mendel discovered this law of independent assortment through a dihybrid cross. You're looking at seed color and seed shape. Got 2 of these different things. And it's a law because as long as they're on different chromosomes, it will work. However, like I said, he got lucky with this because even one of the ones that were actually linked, they just happened to be on opposite ends of the chromosome. So it looked like they were being inherited independently when they really weren't. Now, here's what's important. Even though I say Mendel got lucky, he did not understand the chromosomal basis of inheritance, his experiment was still groundbreaking. He did a manipulated experiment with thousands of pea plants, and he disproved blending inheritance. Think of this. Yellow and blue make green, right? This is important for evolution because in the time when Darwin published Evolution by Natural Selection, it was thought of blending inheritance. So if you got a trait that popped up out of nowhere, it would quickly be swamped out in the population. But Mendel disproved that. He showed that a gene and alleles are discrete things and they can amplify in a population. And that's important that he disproved blending inheritance. That was a huge boon to Darwin's evolution by natural selection. And when we talk about Thomas Hunt Morgan in the next video, he really supported evolution by natural selection, even though he started out not believing in it. So whenever we talk about Mendelian inheritance, what that means is I've got one gene, I've got two alleles. So I've got a gene for flower color. You can be purple or white. One is dominant, one is recessive, and you get one from each parent. Okay, that is Mendelian genetics. So anything beyond that would be an extension of Mendelian genetics, which I will talk about in another video. How many of you have done a Punnett square? Yeah, by Reginald Punnett, right? So what Punnett squares do is they make a prediction of what your outcomes are going to be using Mendelian genetics of one gene, two different alleles. Now, something that I want to talk about. Let's look at these pea flowers. One is white, one is purple. There is a pigment that the purple flower makes called anthocyanin. Cyanin is blue, antho means like plant. And so the purple that you see is a pigment. The white pea plant doesn't produce the purple pigment. So the white allele, that is caused by a mutation. It's a knockout mutation that actually the plant can't produce the pigment anthocyanin, right? That's why if you are heterozygous and you get one functioning copy of the allele, the plant can produce the pigment and it looks purple. That's pretty cool. And anthocyanin, if you like blueberries and blackberries, all these dark colored berries, anthocyanin is in there. It's an antioxidant. It's very healthy for you. Okay, well, this has been another episode of Tom Kennedy Science, and I hope you enjoyed it. And stay tuned and learn about Thomas Hunt Morgan and why I think he's really the real father of modern genetics. All right, Mendel was good, though. Until next time.